Hi, my name is Clemens Falker-Gieske. I'm a postdoc at the Göttingen University in Germany, and today I want to present my data about a mutation that we discovered in white egg-laying chickens that might give them a resistance to amyloid arthropathy. Amyloid arthropathy is a disease which is characterized by swollen knees in affected chickens. If you take a closer look at the knee joints, you will find these orange amyloid depositions, which mainly consist of aggregated serum amyloid A. In the field, the disease has only been observed in brown egg-laying chickens, and white egg-laying chickens are resistant. It is currently thought that a difference in the immune response is responsible for the difference between the two chicken lines, but the genetic component is so far unknown. So SAA is an acute phase protein, and its expression is induced up to 1,000-fold upon bacterial infection or injury, for example. After its production in the liver, SAA associates with high-density lipoprotein and then travels to the site of infection, where it has downstream effects on inflammation and the immune response. So we were interested if the two chicken lines, brown and white layers, differ in their SAA gene sequence. So Professor Simjana provided us with 25 whole genomes of brown and white layered chickens, and we calculated the FST values for chromosome 5, on which the SAA gene is located. The average FST for the whole chromosome was 0.355, which is indicated by the dashed line, and we found lots of variants above an FST of 0.8. Among those was one missense variant, which had the highest FST value of all variants. And this mutation was present only in white egg-laying chickens, with 19 chickens being homozygous, 3 chickens heterozygous, and 3 chickens did not carry the allele. So we were interested in the impact of this mutation on chicken cells. So we overexpressed the SAA wild type and the SAR90S isoform in chicken hepatocellular carcinoma, or LMH cells, and qPCR revealed that the SAA wild type cells express slightly higher amounts of the gene, as you can see here, but the difference wasn't very big. On the protein level, we saw the complete difference. So we checked SAA expression in cell lysates with a Western blot, and as you can see here, we don't see a strong expression of the wild-type protein, but the SAA R90S protein is highly enriched inside the cells. This is also true for the extracellular space. So in the medium of the cells, we couldn't find any wild-type SAA after 24 hours of incubation, but we found huge amounts of the mutated form. So we were interested in the mechanistics behind that, and we thought probably there's a difference in the aggregation propensity of the two protein isoforms. So on the left, you see the detergent-soluble fraction of cell lysates, which means that the protein that you find there is not aggregated. So we find some of the wild-type protein, and again huge amounts of the mutated SAA isoform. So in the detergent-insoluble fraction, which contains aggregated protein, we didn't find any wild-type isoform, and again huge amounts of the mutant isoform, which most likely means that this mutation leads to a higher aggregation propensity in the serum amyloid A protein. using computer programs, we also predicted that this region is an amyloid hotspot in the mutant protein. We also validated that by staining the cells with an antibody against SAA. As you can see here, there is not much expression detectable in the wild-type cells, but with the mutation we see a spotty diffuse localization of SAA, which is also an indicator of intracellular aggregation in these cells. So we were interested in the downstream effect of this R90S mutation. So we ran RNA-seq on the two cell lines, and one striking difference between the two groups was the general strength of expression. So as you can see here, the log-fold changes in the wild-type protein of affected genes is higher compared to the mutant form. We have less differentially expressed genes in cells that overexpress the mutant form, and we have only a little overlap between the two cell lines, which was very surprising to us. So we wanted to know what mechanistic consequences this might have on the cells. So I did some pathway analysis, and I found out that the wild-type protein has an influence on gene transcription, whereas the mutant form mostly affects pathways related to the membrane and receptors. And regarding the localization of downstream affected genes of the mutant form, we have an enrichment towards the membrane, the cell periphery, and the extracellular space. So we took a closer look on these transcription factors that are affected by the wild-type protein, which you see here in the heat map. And the effect of the mutant form was much lower compared to the wild-type isoform. So we looked up these transcription factors, and most of them, or a large fraction of them, have an influence on cell fate determination. So in last year there was a publication in Cell where it was shown in mice that SAA together with IL-6 leads to a change in the cell fate specification of immune cells, in this case CD4 T cells, towards pathogenic Th17 cells. So we checked if we see an overlap between the differentially expressed genes and in these murine cells, and we saw that 24 genes were commonly differentially expressed in our SAA wild-type cells with the murine SAA-induced CD4 T cells, but we only found an overlap of 8 DE genes with the mutant form. So our final conclusions are that the loss of function in the SAA protein might be causative for the disease resistance, that the intra and extracellular accumulation leads to a disturbance of the immune response of white egg-laying chickens, and that the SAA R90S mutation lacks the ability to regulate transcription factors necessary for cell fate determination, which explains the differential immune response between brown and white layers. We will validate in a suitable experiment model in the next few years, and I'm now open for questions. Thank you.